Hi everyone, my name is Neil Zanis and I'm a market manager at Enchain Labs. I will be your moderator and thank you all for joining us today. We will have a Q&A session at the end of the presentation. To ask questions, all you have to do is click on the Q&A in the Zoom navigation bar, and that's going to be at the top or at the bottom of your screen, and type in your question. When submitting a question, please avoid clicking the anonymous button so we can reach out through email if we aren't able to get to your question. We will, of course, get to as many of your questions as we can. And now I'd like to introduce Ben Nappet. Hey, Ben, how are you doing?
Hi, Niels. I'm good, thanks. How are you doing?
I'm good, thanks. Ben is our market manager for LNP solutions at Enchain Labs. Today, Ben will walk us through how Sunbather will help to seamlessly transition preclinical development of LNPs into GMP production for clinical trials and beyond. And so now I will gladly hand it over to Ben.
Thanks very much, Niels. Hello, everybody. Thank you very much, as Niels said, for taking some time out of your day to join us. I'm going to talk today about how our new Sunbather products can help you transition seamlessly into a GMP environment. So when you go out to out on holiday or go to the beach, there's a certain order to how you do things. First you're going to apply your sunscreen carefully, then you're going to go and find that perfect patch of sunshine, and after that you settle down for sunbathing. Now, this isn't the sunbather that I'm here to talk to you about today, but over the course of this talk I'm going to talk you through how the Sunny Suite follows that same pattern to help you transition your development of mRNA LNP therapeutics from screening to GMP. So this is a diagram that many people on this call will be familiar with. It is the outline of the process for an mRNA LNP drug development, going from preclinical development, during which we seek to screen our formulation components to identify a suitable mixture of components and lipids in order to effectively encapsulate, deliver, and release our mRNA. We are looking then to optimize the production process of that formulation that we identify as successful, so that we make the perfect particle every time. And then we're going to look to produce larger volume of those particles in order to do further downstream testing and validate those particle samples ahead of moving towards clinical trials. Once we reach clinical trials, obviously, we're going to now start considering injecting these materials into humans. And as we move through those phases of clinical trials and ultimately into commercial manufacturing, the populations of humans we inject with these materials increases, increasing the risk levels and the requirement for significant regulatory control over the production of these particles, which is why at this point you're going to enter GMP production to ensure that the manufacture of those particles is subject to the rules and regulations that ensure safe manufacturing practices. Now, each of these different steps of development has a different set of ultimate goals, requirements, may have multiple stages of development, possibly be happening in entirely different laboratories. And so there is a need to be able to cohesively join together these different stages of development to ensure smooth transitions between these stages. Particularly when you look at GMP equipment available on the market, there is certainly of late has been a very high focus on large-scale production, certainly with the development of vaccines, producing significant quantities of product in order to be able to deliver to entire global populations of humans. So that typically comes along with having to change your pumping technology and also potentially the geometry of your mixer that you're using to produce the particles. And so implicitly involves re-optimization and redevelopment of that process at each individual stage when you change up that scale. Additionally, when you're first entering clinical trials, the use of single-use flow paths at these high volumes can be a very expensive prospect and potentially a challenge as you transition into GMP. Often when you start off entering clinical trials, the realistic requirement for volumes of production is anywhere between a few hundred milliliters to a few liters or tens of liters of LNPs. And that's enough to get through the smaller populations of doses that need to be generated at early stages. as well as then for some applications that are more niche in target audiences, the early manufacturing stages as well. It's desirable to have an easy transfer from preclinical equipment into that clinical environment, into a GMP manufacturing process, in order to keep that mixing consistent and enable that transition to be smooth without the need for consuming large quantities of expensive material during reoptimization. In addition, it can be beneficial for the flow path that you're using to be flexible in its use, fully replaceable, but also potentially reusable to avoid having high replacement costs during early stage development. Here at Unchained, we don't feel that the GMP transition should be a hard one for you, so I'm looking to make life easy with Sunbather. Sunbather is our GMP-ready LNP producer. It has multiple channels available to pump up to 1.8 litres an hour with our standard consumable flow paths. It has inline dilution factored in with a fully replaceable flow path that can also be validated for reuse if you wish. We offer a range of different glass microfluidic mixer types suitable for use in a GMP environment that can be selected for whatever your application may be. And also with software that offers 21 CFR Part 11 compliance and audit trails internally. So that ensures that you have all the documented information you need about your manufacturing process. We are also able to directly transfer the processes you've developed on our R&D platforms, Sunscreen and Sunshine, onto the Sunbather instrument. Sunnies are microfluidic mixers, come in the same range of flexible geometries as available again on our R&D platforms. Any of these Sunnies can be used on any instrument stage of development. They've been redesigned and optimized to have top connections for the fluidic inputs now, so that even the manufacturing process is even cleaner than it ever was before, and allows for seamless interchanging of the different mixer geometries. These are also then validated and suitable for GMP use with traceability and batch records to boot. Depending on your mixing process, you may choose something like a Sunny Trident, which has a reverse angle mixing geometry and an inline dilution available on the same mixing device. Or alternatively, for a more traditional cross or T-type mixing device, we have the Sunny X and T formats, which come in multiple different channel sizes. So again, as you optimize your process into continuous flow using the Sunshine instrument, you're able to then identify the best mixer available to form your particles ready to transition to Sunbather. Flow path provided with the instrument is flexible. There are two different flow path types available, depending on whether you've optimized your process to involve one Sunny or two Sunnies. So any process you've developed on previous instruments can be easily implemented onto Sunbather. All of the materials used to construct that wetted flow path are fully traceable with material certification documents available, and extractables, bioburden, and endotoxin data provided too. You can therefore choose to reuse the flow path and clean it in place, or choose to replace the entire flow path if you require. And here at Unchained Labs, we are GMP ready when you are. So we're able to offer full audit support should you wish to conduct a supplier audit ahead of purchasing an instrument. We offer an IQ/OQ at point of installation to ensure everything is set up correctly, running smoothly, and the flow rates are validated, and also offer comprehensive user training at point of install to make sure that you're up and running with the system as quickly as possible. With the 21 CFR Part 11 compliance within the software, material certification documentation available, and the entire development of this product, along with the manufacture of this product in Unchained Labs being underpinned by our quality management system, we have everything needed to support you in implementing the Sunbather in your GMP facility. User-friendly software has been designed to work on a touchscreen interface. So the system is provided with a kiosk-type IP66 rated touchscreen PC operating in a kiosk mode. The software has a compliant audit trail that can be readily exported, and operator and administrator access tiers. What you can see in front of you is a screenshot of the operator access. To set up a run, simply select the channels that you wish to utilize, enter the pumping flow rates and a total volume for the production run that you'd like to do. You can enter your own batch ID here, and also a run ID is then generated by the software as part of the audit trail compliance. You can initialize your pumps and then click start to begin your production run. It's as simple as that. Every time you conduct a process critical action, such as starting a run, completing a run, or aborting a run, the software will prompt you for a password so that the action is logged and signed off within the audit trail. This means that you can now go all the way from your initial high throughput screening of a wide range of formulation variations using our sunscreen instrument. Now, the sunscreen instrument allows you to run 96 experiments within 6 hours, a rate of around 3.5 minutes per experiment, fully automated, so that you can set up a plate of experiments, walk away, and come back to a full plate of prepared microfluidically precise LNP formulations. And then you can run a characterization of those all within an hour or two on the stunner as well. So the data you can see here shows a screen of 32 different formulations where we varied the type of pegylated lipid, we varied the cationic lipid used, we varied the helper lipid used, and also the PEG lipid mole ratio, all across these 32 experiments, which were conducted in triplicate, to produce a very comprehensive data set very quickly, which can be analyzed for insights to direct you towards the perfect particle formulation for your application. Once you've identified those successful candidates that you're looking to take forward to optimize for a manufacturing process, you can use the Sunshine instrument, which allows you to perfect your process and hone in on the perfect set of process conditions in order to make the particles the most efficiently and effectively. You can optimize the mixer type, the total flow rates to target things like particle size and PDI, and also from your downstream in vivo and in vitro testing, optimize for transfection performance of your product. Once you've found a perfect set of operating parameters, you can then use that same instrument to transition into a continuous production mode, where you can then produce particles in a continuous flow regime and optimize to ensure that over the production runs that you plan to conduct in a GMP environment, that you're getting stable, high quality particles out of the end of your process. A continuous process is identical to the one run using our Sunbather instrument. We have the same type of pumping channels and the same mixer geometries available to ensure that your transition into GMP production of particles is as seamless as possible. And if you need more production volume, Sunbathers can easily be numbered up to produce increased volumes of product by using the same validated continuous process that you've been developing all the time. That hopefully shows you how the Sunny Suite is able to take your process all the way through that product development process, from screening, optimization, and through to clinical trials and beyond, using the Sunny Suite. From 96 formulations per run at only a few hundred microliters per experiment, through to optimizing your process conditions at a few milliliters per experiment, and onwards to continuous production of liters of experiment, with flexible mixing geometries and automation that saves you both money, time, and also materials by being efficient about how we consume your valuable reagents. You can ensure that by using the same process and same mixing throughout, you're producing the same high quality particles end to end with the Sunny Suite. Some data to back that up, we can see that when we run our sunscreen in its low volume protocol mode, our Sunshine system in both its protocol and continuous modes, and also Sunbather in a continuous production run, that we get the same size particles with the same low PDIs when we run the systems with the same process conditions and the same mixers. Here at Unchained, we're very excited for the Sunny Suite instrument to join the family, the Sunbather instrument, to join the Sunny Suite family, and look forward to answering any questions that you might have about these instruments. Thank you very much.
Thank you, Ben, for taking us through how to use the Sunny Suite to take LNP formulations right through from screening and process optimization, and now into the GMP production with the Sunbather instrument. We already have some great questions that have been submitted. You can, of course, still ask a question by entering it in the Q&A section of your Zoom navigation bar. Okay, so let's start with the first one. Do I need to change the sunny every time I reuse the tubing kit?
So you can change the sunny when you reuse the tubing kit. So the sunny is typically the narrowest constriction within the flow path. And so you may consider it good practice to simply swap that mixer out between uses of the system, but there is no obligation to do so. It can be cleaned in place along with the rest of the flow path if you wish.
Okay, and how do I clean the flow path?
So we've done testing and validation of a cleaning process that we provide our customers with as an SOP when purchasing the system. But this can be tuned and adapted to your own requirements. But overall, the cleaning process itself takes around about an hour.
Okay. In the same line, someone's asking how long it would take to assemble the flow path or the overall setup.
Sure. So the flow path is delivered in a partially assembled state in a double bagged pack. And so it takes around about 20 minutes to half an hour to assemble onto the system conservatively. And so there's another recipe that helps to provide customers with information on how to do that assembly. You don't need anybody to do that on your behalf. It's very easy to use or assemble. The only tool required is a torque wrench that's provided with the system as well.
Okay, thank you. A longer one. Does my process transfer directly from a Sunshine protocol mode into the Sunbather, or would someone have to run Sunshine in a continuous mode?
So the way that our systems work at the R&D stage with Sunscreen, what we're doing with the system is releasing small volumes of your reagent into an existing flow path of a driving fluid, which is normally water and ethanol. And in the little video you saw in the slides, you could see we did an experiment with some dye solutions, and those represent your sample going through that flow path. So when those systems run in this protocol mode with small volumes, we can typically take a head and tail cut to collect just the central portion of the sample, which is going to be the most representative of a continuous flow experiment. So it is possible to take those results that you collect from that system and directly go into continuous flow production of your samples. But normally on Sunshine, there would be a little bit of optimization as you transfer from these small volume experiments to a longer production run in continuous mode. And so you'd simply have to undergo that development process using the Sunbather instrument. But yes, so it would be possible to go directly from one instrument to the other.
Okay, great. Which is practically similar to the next question, but from Sunscreen. Can someone go directly from Sunscreen to Sunbather?
Yes. So as that sort of, I think I kind of answered that question by accident in the previous one. So yeah, you can do that and transfer directly across to the Sunbather instrument. You just need to do a little bit of optimization as you transition to continuous flow and run larger volumes through the system. If you're using Sunshine in between, you can optimize those flow rates and run durations outside of a GMP environment, and then port that protocol directly into GMP.
Okay, great. That's clear. Now, when running the Sunbathers, like you said on the end, you can have multiple. So if you would run them in parallel, would they all be connected to like one computer so they can all be started at once, or would that be separate runs?
So right at this moment at launch, they would be separate, but we're very willing to discuss, if somebody wants to run Sunbathers in parallel to have a chat with us, and in the future we'll be able to implement running those all from a single unit. But right now, it's a single kiosk PC that comes with the instrument, and that one kiosk runs each individual device.
Okay. Beyond lipid formulations, to broaden the horizons, will the Sunny Suite also work for the production of other nanosystems, protein or polymer based?
Yeah, absolutely. So Our systems have been used extensively, or certainly the history of our instruments in microfluidics have used all sorts of different nanomaterials, whether that be polymer nanoparticles, different types of lipid particle, lipoplexes, polyplexes, all sorts of different applications. Our validation and testing has mostly been done on these instruments in the LNP space, but absolutely you can use all of these instruments to produce polymer nanoparticles as well, or other types of material that can be produced by a continuous flow process. Typically, it's a solvent and an aqueous solution that trigger a self-assembly or precipitation or controlled precipitation of material to form particles at room temperature. So yes, you definitely can use this for other applications. The materials that have been used to create the flow path are all excellent or good compatibility with the solvents expected to be used in the system.
Okay, great. Those were all the questions that we had for today. So thank you for answering all of them, Ben. And again, thank you for the great presentation. And I also want to thank everyone that joined us live today. If you would like to have a deeper conversation with our team, please do get in touch with us at info@unchainedlabs.com or learn more at our website at www.unchainedlabs.com. We would love to connect with you and talk science. Thank you again for attending our virtual seminar, and I hope everyone has a great rest of their day.
Thank you. Goodbye.